Okay, last talk of the meeting before posters. But last but not least, I have to say that working for a very long time in landlocked Africa, I get a little bit jealous of pictures of Southwest Africa or Southwest Asia Isomer sites. So, without further ado, Chris King, thank you.
Thank you very much for the opportunity to come and talk to you about some of our work. Have we started at the end here? How do I? I think we're at the end. Yes. There we go. Okay, yeah. Perfect. Yes, thank you for the opportunity for coming to speak. And I'm speaking on behalf of Jim Kazura, who's the program director, and he was unable to make it today. He's actually in Papua New Guinea now. So the title of the talk is really, I want to focus just on a few areas that we've worked on in the Isomers. As you know, these comprise many different projects. But this is really want to focus on how this intensified malaria control measures in Papua New Guinea that really was rolled out between 2009 and 2011. And we had the opportunity to capture some of these evidence, this rollout, and how effective this was. And this is really a collaboration between the Papua New Guinea Institute of Medical Research, which has been an area that's done a lot of very good malaria research over the years and has a good, well-dedicated staff, the Walter and Eliza Hall Institute, the Swiss Tropical Research Institute, the Burnett Institute, and Case Western Reserve University. Okay. So what I want to do is just focus on a couple points that I think that are fairly unique or different in the South Pacific. I'd really like to talk about the epidemiology of malaria in Papua New Guinea, and Solomon Islands was one of the other research sites. I'm not going to talk much about that because of time, but just the only point I want to say about the Solomon Islands is that this is a low transmission area. It has almost 14% of Plasmodium vivax and very, very little Plasmodium falciparum. And it's interesting why that still persists. And they're mostly asymptomatic infections, very much like descriptions that you see down in South America. What I'd really like to focus is on the impact of malaria control measures on the burden of malaria, transmission by mosquitoes, and a little discussion about immune correlates of protection. So, like our study sites in many areas, just to highlight, is that the Isomer study design that we did in the Southwest Pacific is that we had two sites in Papua New Guinea, as I mentioned, one in the central province of the Solomon Islands. We did serial cross-sectional studies. We compared this to historical controls with samples that were collected pre-Isomer and also pre-intervention for malaria control. And then we did two post-intervention cross-sectional studies, one cross-sectional study in the Solomon Islands. And importantly, we did these cohort studies of about 500 people. And these were done in each of the study sites for about 40 weeks, doing active surveillance, very much like the studies were described in Uganda, where we monitored people every 2 weeks, both active surveillance and passive surveillance for clinical disease. So there's quite a bit of information in those. We also did two other things. Mainly, we monitored malaria with the PNG National Department of Health Sentinel sites, and I'll describe that later, to look at the overall impact of malaria transmission and prevalence throughout Papua New Guinea with these malaria control measures, and we helped the surveillance with the molecular diagnosis of malaria. And we also started passive monitoring for severe malaria in several PNG district hospitals. So here's an idea of what malaria looks like. I think Papua New Guinea is unique. The Solomon Islands is back over here. This is island chain here. And the Solomon Islands would be right down past, this is here, and the Solomon Islands is right down here, so it's all in the South Pacific. What you see here is this is falciparum, and this is vivax. They're sympatric. I think the really remarkable thing about Papua New Guinea is that the intensity of transmission, if you look at entomologic inoculation rates, are almost identical between vivax and falciparum. You also find Plasmodium malariae and Plasmodium ovale fairly frequently. So the other interesting thing is that probably Plasmodium vivax has got one of the higher intensity of transmission throughout the area. You see these brown areas across the center part of the island there. And this represents highland regions here. And these are areas where it's probably over a mile high, or about 1,500 meters. And these are too cold for malaria transmission to occur. although they're warming in Papua New Guinea, is actually you're seeing increased evidence of malaria transmission on the highlands. You can also see vivax tends to get up higher into the highlands in some areas. You see if you're about 13 to 11 or 1,200, about 1,300 meters, you see almost solely Plasmodium vivax transmission up there, and there's a fair amount up there. So one of the things I want to focus on is some of the transmission similar between vivax. Acquired immunity, I want to point out, to vivax occurs much more quickly in Plasmodium falciparum, and this is sort of interesting. And then the relapses contribute to a large bulk of P. vivax infections and likely development of acquired immunity. So here are some cross-sectional studies that were done, some historical studies that we did pre-malaria interventions. And as you can see on the left side of the graph, you see in the red represents the incidence of clinical episodes of vivax or falciparum malaria per child per year. And these are spanning from the ages of about 1 year to about 4 years of age. And as you can see, the dominant infections in the early children are really vivax. And this is what you see is the primary cause of clinical malaria in Papua New Guinea in children under 2 years of age is really attributable to vivax. And then falciparum malaria tends to rise and then peaks at a much later age. And this is what you see in many of the areas throughout Papua New Guinea. And interestingly, as you get into about 4 years of age, you see the instance of clinical cases of vivax malaria really drops, although the number of infections still persists. And this highlights in the right-sided graph where you look at individuals, and this is a cohort study that was done where every group of over 200 children, I think 206 children, were treated, and then treated with malaria, collected samples, and then followed every 2 weeks for the presence of infection by PCR, by blood smear, and then evidence of clinical malaria. And you can see here is that every child became infected over the period of follow-up. In fact, there are on the average of 5 separate genotypes of malaria occurred both for P. vivax and P. falciparum over that period of time, virtually identical rates of infection. However, about half the children actually acquired clinical falciparum malaria, whereas vivax malaria, there's only a couple cases. And so you really have very little vivax malaria, even though you have a lot of infections going on. So this is interesting how that acquires quickly. Seen in other areas. So I wanted to show this slide because one of the reasons that we think under the similar transmission conditions that you get vivax develops acquired immunity or you have very little clinical cases develops very quickly is that they have a lot of relapses. So we always talk about relapse, but how much do relapses really contribute to the blood stage infections, and what are the evidence that demonstrates this? This is not an Isomer-supported study, but another study that was supported elsewhere and also supported by the National Department of Health, looking at a study where they did a cohort study in a very similar design as I described previously, starting out in the upper left-hand panel where they took children between 5 and 10 years of age, and this is a study that was recently published, and they randomized and they received coartem to clear the blood stage infections, and then some children were given primaquine, 0.25 milligrams per kilogram up to 14 days by direct observation, so we know they got them, and the other group got placebo. And then what you can see is that you monitor the number of clinical cases of both in the placebo versus the primaquine, and you can see that particularly within the first 80 or 3 months, you see a dramatic increase in the number of vivax infections occurring in the placebo group compared to the control group, suggesting that the vast majority of these things are contributed by the relapsing parasites. You continue to see more, but the majority is occurring within that first early area. And what they did is that we went ahead and we sequenced and actually looked at the genetic markers of trying to identify unique parasites. And you can see that in the group in the control, that there are about an average of 5 unique parasites occurring within the 8 months of periods compared to about 1.6. So the point is that about 4 out of 5 or 3 out of 4 new infections within the first 3 or 4 months of vivax were attributable to relapsing parasites. So this provides us an opportunity to really look at some of the genetic markers that one can do this without doing the primaquine interventions. So that's probably a major cause of this. The other thing I wanted to show is actually we looked at gametocytes and trying to understand the role of gametocytes in some of the transmission as well as some of the, and what are some of the factors that drive levels of gametocytes. This is a little bit complicated graph, but it does give you an idea of the epidemiology in Papua New Guinea, cross-sectional studies done. And if you can look up in the upper left-hand panel, shows falciparum, and this is a fairly typical pattern that's seen in some areas of moderate transmission. You can see in the dark sort of lines there with the PCR, I guess that's purple, not purple, sort of dark brown. Let's see here. And you can see that the prevalence of malaria across the age group actually rises for falciparum, peaks at around 9 to 12 years of age, which is fairly typical. And then you can see by blood smear it's higher earlier. And you can see the parasite density is much higher of those people that were children that were positive for falciparum, much higher. And as you develop acquired immunity, the intensity of infections drop. And what you can look at, if you look in the lower graph, we actually looked at gametocytes, both by blood smear and by PCR, looking at PFS or PFVS25 by taking 50 microliters of blood from individuals, putting directly in RNA later, and later amplifying, reverse transcribing it to look at the gene expressing PFS25. And I'll show you data a little later that correlates very well with the gametocytes. And as you can see, that the presence of parasites and the frequency correlates pretty well with the frequency of gametocytes. So if you've got blood smear positivity, you're going to have gametocytes. And that's pretty much what other people have shown. But importantly, what you see is you see the gametocytes are much higher when you have higher parasitemias. And I think, and you see the same relationship with Plasmodium vivax. So the point here is that when you actually correlated the burden by PCR of malaria infections for both Vivax and for falciparum, more striking for Vivax, that the relationship of being gametocyte positive and is directly proportional to the level of asexual parasitemia. And over on the left-hand side, you can see a correlation of our transcripts for PFS25. So we got about 10 transcripts for every blood stage gametocytes by blood smear. So I think the important point there is that you get gametocytes at very low levels, their presence, but the level of gametocytes and likely the ability to transmit is very much related to the level of parasitemia. So if you can suppress parasitemias, get very low levels, probably the ability to transmit from gametocytes is probably reduced. So the other thing is I really want to focus now on a little bit of work that was done about what are the impact of transmission and interventions in Papua New Guinea. And here's again a picture of Papua New Guinea. The research sites that we did for the Isomers are in red. The dark blue represents the Sentinel sites done by the National Department of Health that actually did both looking at vector biology, and we also did vector work in our study sites as well, monitoring what are the vectors. And also they measured the frequency of infections. In each of these different areas, they looked at clinical disease, a number of different parameters. They also monitored bed net use. So in Papua New Guinea, like in many places, the introduction of control was rolled out by the World Bank, and there wasn't much of the President's Malaria Initiative there. And it's the same sort of thing that we've talked about in Africa in many areas. They're using long-lasting insecticide-treated bed nets. One thing is that there's no pyrethroid resistance, or very little, in Papua New Guinea, probably because they don't use very many treatment of agricultural like this needs. We also did introduce the rapid diagnostic test, so rather than people coming with clinical syndromes of malaria, they're tested with RDT before they're treated. Coartem was used for the first line of treatment, and then did intermittent presumptive therapy with sulfadoxine pyrimethamine in pregnant women, as was described. And so what you can see down here is the uptake of bed net use across these different Sentinel sites and also in our particular study areas. And as you can see, starting from 2009 to 2011 was the major distribution. Another round of distribution occurred in 2014. I think a lot of other people can see is that the uptake was not that great, with about 50% of people, 60%. And then after the 2014, still bed net distribution is still pretty not very effective and hasn't really accelerated. And probably what that really relates to is that just the simple remoteness of many areas in Papua New Guinea. They're just hard to get to, hard to get the bed nets, and also the number of bed nets available. And it's just very weak infrastructure in Papua New Guinea in delivering things by the National Department of Health. But however, they've had an impact, and a pretty striking impact. And what you've seen is that here is look at some of the Sentinel villages. So our two study sites was an area called the North Coast, or Madang. And it's interesting, we talked about Edward Koch recently. I think somebody brought him up. And that's actually where Edward Koch worked back in 1900 in a place called Alexishaven. So we can see some of those old places there. So this is some of the same area that he worked over a century ago. And you can see these are the entomologic inoculation rates. These are some of the Sentinel villages in this area, both on the coast and inland, and that's important because the vector species differ. And you can see the year before introduction of bed nets, and you can see again, here's both falciparum and vivax if you look at these things, and this is done by the circumsporozoite protein. And then after bed net treatment, boom, it dropped. And it continues to be very low in some of these areas. Down here, in an area with really high Yawatung, you're getting 1,000 EIR, so there's areas where you can match what you get in Uganda in isolated places. And again, you see a drop, and it's coming up a little bit, and now the same data suggests it's rising up a little bit, but it's still significantly reduced what it had been. And this has also been accompanied by really a dramatic decrease in the overall intensity of malaria infections throughout Papua New Guinea, and these are for the whole data. And you can see the overall prevalence of any species. Plasmodium falciparum and Plasmodium vivax have dropped over this period of time from 2009 to 2014, and particularly in the target population of prevalence. And this also is accompanied by a drop in this, cases of clinical and severe clinical malaria as well. So they've had a significant impact. However, this impact has not been uniform. Some areas have shown a tremendous effect, and other areas have shown much less effect. And to try to understand what are some of those reasons why some of these impacts have worked in some areas and not in others has been part of our interest in our Isomer. So to focus in on one of the study sites, and this is in the East Sepik province up here, and this is actually up here, there's a mountain range along here, a big Sepik River going on here and along, and then this is a very area of quite high population density and quite high transmission of malaria in some of these areas, both vivax and falciparum. So what we saw, and when we did some of these cross-sectional studies, as you can see in 2005, before any interventions, you're seeing about 55% Plasmodium falciparum, 35% vivax. The vast majority are asymptomatic when you do these cross-sectional surveys. Again, these questions, how many of these actually become symptomatic later? We did a little bit of that monitoring, and we found very similar results. About 20% end up becoming ill later. Many high frequency of submicroscopic infections. By the time you get 2012, 2013, after the interventions, there's a dramatic drop. You get a reported 95% bed net use, how much are actually being used, and monitoring how well they're used. It's hard to evaluate, but they basically, it's like we're talking about Uganda, there's probably about 1 bed net per household. And that's probably not very much considering the number of people using, so I doubt they're going to have a big impact. But certainly, there's a dramatic drop. And it's seen elsewhere. And also the multiplicity of infections was also decreasing. And as you can see, both for falciparum and Plasmodium vivax, if you look at the gray bar on the right side there, that's more than 3 or 2 parasites found simultaneously. And you can see the multiplicity of infection for falciparum dropped after the bed net use over a period of several years. And you saw the same thing for Plasmodium vivax. So these are indicating that there is a significant drop in transmission and level of infections. So, and actually, and here is sort of mapping when you start looking at these villages for in areas where it had an impact. If you look on the left-hand side, represents in 2005, and on the right-hand side, 2012, 2013. The blue represents Plasmodium falciparum, the red Plasmodium vivax. And you can see that the size of those little circles is represented to the proportion of individuals or the frequency of malaria infections. You can see if you go from 2005 to 2013, there's a marked drop. However, it's variable. So there's certain areas, if you go into this village up here, you can see Sunulu, that there's still quite a bit of falciparum in there, quite a bit of vivax. And if you go and actually explore in those villages, what you see is reduced bed net distributions, but you also see differences in the vectors. So we went out and started doing vector collections. And the 2 major complexes of transmitting vectors in Papua New Guinea is Anopheles punctulatus group and Anopheles ferraudi group. And it turns out they have very different sort of biting behaviors. We find the ferraudi groups, depending on the times, often more in the coastal areas. But before, and the punctulatus tends to be more of an indoor biting mosquito species. And even this was done in 2013, 2014, but even before 2013, 2014, you find primarily indoor feeding. I'm not showing you this data here, but basically this is done by you have somebody sitting outside the hut and they collect from 6 in the evening to 6 in the morning every hour. They collect mosquitoes. And then somebody inside the households collecting landing catch mosquitoes at that time. So that's what we mean by out versus in. And we've actually done barrier screens as well, showing that there's a whole bunch of other mosquitoes that are not necessarily there in the villages. So this Anopheles punctulatus has got a mixture of both indoor and outdoor biting across here. And this has actually been changing. You actually saw in pre-intervention, you would see predominantly indoor biting. So what we're seeing is that in some of the areas is that you're seeing a lot of fairly outdoor biting mosquitoes in Papua New Guinea, and I think this is what limits the ability of some of these nets to work as effectively. And this varies. The other study site is in Madang province, as I mentioned before. And up here are the Sentinel sites here, and again, and these are our study sites. And again, the cross-sectional surveys show, again, from 2006, 2010, and 2014, a dramatic drop, not quite as much as we saw in the previous study sites. And as you can see, again, they're mostly asymptomatic, and then as you're going down, you get more symptomatic infections. So what we're seeing down here is, again, is somewhat drop, but not as great. And again, what we think this is going here is when you look at these mosquitoes, the major dominant mosquito group here is actually Anopheles ferrati. About 90% of the infections are Anopheles ferrati. And if you can look here, you can see they're mainly outdoor biting mosquitoes. Whereas even in some of the punctulatus groups here, you're actually getting a fair amount of outdoor biting. So what we think is that some of the reason that we're not seeing as much of a drop there is because you really have more of an outdoor biting mosquitoes. And I don't have show data here either, but you see that the selection pressure is quite great. So I think what we're really seeing is that a combination of bed net distributions, difference in anopheline vectors, and a lot of outdoor biting behavior that can be rapidly selected for is really limiting the effectiveness of some of these bed net interventions. Although once they work, they're highly effective. And I remember collecting mosquitoes for lymphatic filariasis, and we went in just before the introduction of these things, we get 200 mosquitoes a night, no problem. As soon as we came back, I could hardly find any anopheline mosquitoes. It was really quite striking. And I didn't get bit as much. So they're quite effective in doing it. So I want to summarize in simply saying is that there is good progress in reduction, but it's not met the targets. You need better bed net coverage. You need better distribution. And I think it's certainly less effective against outdoor biting mosquitoes, primarily those areas that have Anopheles ferrati. And then there are greater reductions in areas, particularly East Sepik province, and particularly in those villages that seem to have more of the Anopheles punctulatus group. So one of the things about the reduction that was talked about before a number of times by Brian Greenwood and others that shows that when you actually reduce the prevalence of malaria, you begin to get increased frequency of symptomatic infections. And this is very much what you see in Papua New Guinea. You can see before the introductions, you see this prevalence curve high for falciparum, vivax peaking more earlier. And then yet again, this is the portion of people that are actually infected with malaria that are actually symptomatic. And what you see, this proportion is now increased after interventions, both for falciparum and for vivax. So why is this increasing? Obviously, it's probably loss of acquired immunity. How does it occur uniformly across all individuals, and how do we monitor that naturally acquired immunity? So because of time, I'm not going to go into any detail on this, but we've been really interested in looking at how do we measure these measures of acquired immunity, and what are some of the markers that we can look at this, and what are the markers to use, and can we use these things to predict which group populations and individuals may have lost their acquired immunity and may be more susceptible to clinical malaria. And so two of the approaches we're really looking at is the breadth and level of antibody responses to multiple malaria antigens and also functional antibodies. The focus is really, acquired immunity can go in many areas, and we've talked about a number of these things, but our real focus has been really on blood stage infections, in part because this is what drives clinical disease, and is primarily humoral, and it's a little easier to measure humoral responses than it is to measure cellular responses that may be more important in pre-risk stage immunity. And clearly, as you well know, is in high transmission, you reach a certain threshold of antibody responses. And is there one antibody or there's multiple immune responses that are required to that? Obviously, as transmission goes down, the threshold drops and presumably makes people more susceptible. So if we can monitor what these thresholds are, and looking at this would be a valuable tool in trying to understand what populations are susceptible to malaria. And some of our studies is really showing is that antibodies to PFEMP1 on the surface of infected red cells for falciparum seems to rise very early and correlates with clinical immunity quite quickly. It's very hard to monitor this because it's so diverse. You have to look at a lot of different antigens. But that's clearly the case. The merozoite surface proteins have been another marker, tend to occur later, and we're beginning to develop certain malaria-specific antigens. So how do you monitor these things? You've seen a number of pictures of the protein array chips that were introduced by Phil Felcher. These are very attractive because you can use very small amounts of sera, literally 1 or 2 microliters, to look at this. You can get a huge representation of what the antigens are doing in it. The problem is that a lot of these proteins are not properly folded, may not recognize the critical portions, and they're expensive. Although Phil has really come up with a new method now where you can actually take the recombinant protein, stick it on a chip, and using a portable, different mechanism to actually measure the protein arrays using on the surface of these chips. And so we're testing those now that could be deployed easily. We use a bead array system, things like that. We've also explored functional antibodies like blocking antibodies to Duffy binding protein in dark for vivax malaria, which correlates nicely with protection. So anyway, what we've found initial findings is that, and I took out these slides because of time, but that the breadth and level of antibodies to PFE1 and blood stage antigens drop dramatically after these control interventions and correlates with increased risks of clinical disease. There are a few people that do not drop these things level and seem to be relatively resistant in our longitudinal cohort studies. And some of these binding inhibitory antibodies, Plasmodium vivax, Duffy binding protein, correlate nicely with protection against vivax malaria. However, many people develop get good clinical immunity to Plasmodium vivax without having these high blocking antibodies. So I just want to summarize in some of the points I've tried to make is that malaria control measures are working in Papua New Guinea but are not meeting national targets. So I think they've got the tools if they can roll them out effectively and understand the vector biology. That doesn't seem to be the case in Africa. We need to know a lot more about the biting behavior in different areas and how this might affect the interventions and coming up with other novel ideas about screens and bed nets that Tom Burcott's done and others have done in the Solomon Islands where they put these barrier screens and having insecticide pregnated and whether this can reduce transmission in areas. And that data is still coming out now, whether that's going to be effective. And certainly as we learn more and more about the correlates of clinical immunity, this can give us another tool to better understand what populations are highly susceptible to malaria as we drop transmission. So anyway, this is, as many of other people have mentioned, this is really efforts by a large number of people, particularly the Papua New Guinea Institute of Medical Research. I've not put the Solomon Islands folks on here, but they're critical people here who've spent many long times and have done all this field work that's been done out here. So there are many, many people. It's been a good group. It's been a pleasure to work with them. Thank you very much.
We have time for questions.
Okay.
Thank you. I'll hand it over to Peter.
Well, I'd just like to express thanks, I think on behalf of all of us, to the 10 Isomer speakers who gave exceptional lectures, really gave an amazing perspective of worldwide malaria. I'd also like to thank Brian for his wonderful keynote lecture, the National Institutes of Arthritis and Infectious Disease for the support of the Isomers, and Michael Bloomberg and the Bloomberg Philanthropies for the support of today's program. When you go to the reception, which I hope you will do shortly, you'll probably see Trish Ward and Genevieve Williams. And if you do, please express your thanks, because they're the organizers that put all this together. We had a lot of the fun, but there were many days and weeks of effort on their part. So with that, let me adjourn the meeting and launch the World Malaria Day Beer and Wine Reception in Finestone Hall. Thank you.